Okay, I'd like to thank the organizers for giving me the opportunity to speak to the die-hard core of conference attendees who are staying to the hopefully not so bitter end. So many of us are here at this conference because we're interested in the remarkably specific patterns of gene expression that are central to life. And one of the fundamental questions that we have to answer is, how is that specificity achieved? How do genomes, and large genomes in particular, encode the specificity necessary for gene regulation? So an illustration of the problem we face is the transcription factor that has to find its binding site in the middle of a vast excess of alternative binding sites. You've already heard about this today. So I'm going to give you an example of the transcription factor CRX, which is a photoreceptor specific transcription factor, and not surprisingly, it's the one I'm going to discuss for the rest of this talk. CRX recognizes this short degenerate motif. It's been characterized in vitro and in vivo by my collaborator Joe Corbo and his colleagues. And as you heard from Iris and others earlier, this story before, if you scan the genome for copies of this motif that are above an affinity threshold that is actually well above the affinity of the known weakest CRX sites, you'll find about 6.6 million copies of these CRX motifs in the mouse genome. What fraction of them are bound? Well, Iris showed you the same number before. 0.2% of these motifs are bound, about 14,000. They fall under about 5,500 ChIP peaks. So the question is, where is the information coming from to direct CRX to its binding sites in the genome? So in the literature, there are two broad classes of models that are often invoked to explain this. And I'm going to treat them as distinct models, but as you'll see, they're not really necessarily mutually exclusive. The first model is the extended chromatin context model. This model says that CRX is directed to its binding sites in the genome by the larger chromatin context. And so the reason why most of those 6 million copies of the CRX motifs are unbound is because they remain in closed chromatin. An alternative model, but as I said, not necessarily mutually exclusive, is the locally encoded sequence grammar model. As I showed you, a motif by itself is not enough to encode binding in the genome, but there could be other sequence features. You've been hearing about some of that today. I'm showing you a made-up example here, perhaps. When you have two motifs in a favorable orientation, then you get binding. So these two models make different predictions. So this first model says that if you take these unbound motifs and remove them from their genomic context and assay their function, then you shouldn't see a difference between these two, these bound and unbound motifs. That function is largely due to the chromatin context. On the other hand, model 2 says that even if you remove these motif occurrences from their position in the genome, you'll still see a difference in function. So I set out to test these two models using one of these new massively parallel reporter assays that's coming online. The version that's been developed in the Cohen lab is called Cis-regulatory analysis by sequencing, or CRE-seq. In this assay, we use array-based synthesis to synthesize sequence elements, cis-regulatory elements. In this case, it's going to be sequences from CRX ChIP-seq peaks. These synthesized elements are cloned upstream of a minimal promoter. In my case, it's going to be the minimal promoter for rhodopsin, so it's a fairly robust, active photoreceptor-specific promoter. So these elements, they're cloned upstream of a minimal promoter, a reporter gene, and the reporter gene is tagged by a unique sequence barcode. So we transfect this library of plasmids into whole retina, and then we sequence the barcodes to get the expression measurement of the reporter gene, and then we normalize that expression measurement by the representation of the barcode in the original plasmid pool. So just in a little more detail, what I want to emphasize, what's going into this reporter assay. Because we synthesize our sequences, they're very short. The sequences I'm going to tell you about today are only 84 base pairs taken from the centers of CRX ChIP-seq peaks, and they are on plasmids. They've been removed from their genomic context and placed in a more permissive environment. So I'm going to move ahead and test these two models by assaying CRX ChIP-seq peaks. So this is what I put into the library. I took about 1,300 CRX ChIP peaks, including bound regions, ChIP peaks that had CRX motifs. And as a control, I also had a matching set of regions where the motif was abolished by a point mutation. I also included bound regions from ChIP peaks that did not have a recognizable CRX motif. If you're familiar with ChIP, you know that many peaks do not have a recognizable motif for that transcription factor underneath them. This is an interesting story, but I'm not going to tell you any more about these bound regions without motifs. I'm going to focus on these. I also, as control sequences to compare, I included unbound regions that had CRX motifs. And these regions were chosen so that their motif content, their CRX motif content, matched what was in the bound regions. And then finally, as a negative control to get baseline activity in our assay, I took these sequences and I scrambled them. I created scrambled sequences, but I scrambled them in such a way so that I preserved all dinucleotide frequencies. So this is the experimental setup. Let me show you the negative controls first. I'm going to show you the baseline level of activity in our assay. The green distribution shows you the CRE-seq expression values for about 900 negative control scrambled DNA sequences. And what I want you to notice is that there's some variation here, but it's not actually technical variation. In these little TIE fighter things right here, I'm showing you 5 individual scrambled sequences with their 95% confidence interval. And what you can see is that random DNA creates distinct levels of activity in this reporter gene assay. So this is really our empirical null distribution against which we're going to define function. So how do CRX ChIP-seq sequences compare to this? As a class, they show function, more function than random DNA. So you can see there's both more activation, and we actually see some repression here coming from these very short 84 base pair sequences taken from ChIP peaks. And I can show that this activity here depends on CRX, or at least the presence of a CRX site, because when you mutate those CRX sites, you see much of that activity go away. So what we're seeing here is CRX-dependent activity. Function has been recapitulated outside of its genomic context. Now, what about those unbound motifs that were taken from the genome? If you look at them, you see that they look much more like the random DNA. In fact, on this far end, they're pretty similar. We do see some repression. This result suggests that the unbound motifs removed from their genomic context they're intrinsically non-functional. Now, I just want to take a little time and emphasize how striking this result is here. In the blue distribution, every single sequence has at least one high-quality CRX motif, and usually more. The green distribution, you almost never find CRX motifs because they've been scrambled. And clearly, these two distributions are largely falling on top of each other. So this result shows that these unbound motifs seem to be intrinsically non-functional. And looking within the CRX ChIP peaks, I can find sequences that some that act as promoters and others that act as enhancers. So in this first set of data, I showed you what happens when your minimal promoter is a fairly active row minimal promoter. I replaced that with a non-autonomous, basically just a little more than a Tata-box sequence. And there are now some ChIP peak sequences that can drive activity from this very minimal setup. And you get this interesting bimodal peak. Some sequences are off and others are on here. Interestingly, the random sequences show that bimodality as well, and we're not sure what the sequence features are that distinguish those yet. Okay, so let's take this back to the models. So I tested these models by removing unbound and bound sequences from their genomic context and tested their function. And contrary to what model 1 predicted, I saw that there were functional differences between the bound and unbound sequences when they've been removed from their genomic context. So I'm going to say my data, is inconsistent with this chromatin context model. Now, I want to be clear what I'm saying. I'm not denying the reality of, say, position effects or the importance of chromatin in the mechanism of transcription, but what I am arguing is that the distinction between functional and non-functional motif occurrences in the genome is locally encoded in a context-independent manner. That there are encoded sequence features that are distinguishing between all these bound and unbound motifs, and it's not just that the chromatin is rendering 6 million CRX motifs inaccessible. So what are those sequence features? Well, much of the analysis is ongoing right now, but I can say that we have not yet been able to find any other transcription factor motif or particular spacing or orientation of CRX motifs that can explain this difference. But one thing that is a very obvious difference between bound and unbound motifs, as you've been hearing before, is high GC content for the bound motifs. And one question that many of us are interested in is, what is this high GC content doing? The hypothesis that we're pursuing is something you've heard about before, DNA shape in this case. So I've examined the DNA shape of the bound and unbound motif occurrences, and using this program called ORCID2 out of Tom Tolos's lab, we find that CRX chip peaks have wide minor grooves. You can see minor groove width score, wider minor grooves is higher on the y-axis here. Bound motifs have wide minor grooves, whereas unbound motifs tend to have narrower minor grooves. We also looked at propeller twist, which you've heard about before. This is a preliminary analysis conducted on some of our motifs by Lin Yang and Remo Rowe's lab. What we have here are the aligned motifs, bound motifs and unbound motifs, and the heat map shows you the propeller twist. And really what you need to do is look at the averages here. So there's more blue over here compared to that one. So this suggests that there is an important difference in propeller twist around the functional motifs. So this is an interesting hypothesis. Fortunately, because we synthesize our elements and we test them in our assays, we can test this by trying to redesign these motifs, these unbound motifs. And so I don't have data to show you. That's an ongoing effort right now. But what I would like to do is redesign the sequences flanking the unbound motifs to have weaker propeller twist or wider minor grooves without actually changing the motif itself, and thereby testing the hypothesis that DNA shape contributes to function. All right, so I'm going to sum up 3 main points I want you to take away. Chip peaks are distinguished from unbound motif occurrences by local context-independent features. I took short 84 base pair sequences, removed them from their genomic context, tested them in a plasmid assay, and the functional differences that you observe in the genome were still largely preserved. I've suggested that DNA shape may encode some of this local context-independent function. And I'd also like to hope you take away the value of on these large genomic assays, now that they're becoming more feasible, including large-scale negative controls. This provided, in our case, a very crucial baseline against which we measured function. All right, so I just want to acknowledge the members of the Cohen Lab, especially Jamie Kwasniewski, Ilaria Magno, and Chris Fiore, who've developed the CRE-seq assay and taught me everything I know about it, and my postdoc advisor, Barack Cohen, and my collaborators who did the mouse work, Joe Corbo and his tech, Connie Myers. Thank you very much.
Final talk to the conference. So I have a question about your random motifs. So first of all, they show this by modality. Second, they show all of these shifting, you know, above what you would expect, you know, a non-active sequence to have. I'm wondering if you took any extra steps in making sure that they don't contain motifs, because if you scramble a genomic sequence, and it's a pretty big sequence, you're going to have motifs within there. So you could actually ask, you could actually now do an analysis of your randoms to basically see which ones show high activity and just try to discover the motifs within them. Have you tried that?
Yeah, so I purposely did not remove randomized sequences that might have motifs. I scrambled them in as much of an unbiased way as I could. I have looked to see if there's any one motif or handful of motifs that will explain the high expressing sequences. I haven't found anything yet. So because this expression is mostly in the outer nuclear layer of photoreceptors, I've worked under the assumption that most of the transcriptional activity there is coming from these cell type specific transcription factors.
Oops. And what about the bimodality?
That's my timer. I apologize.
Thank you.
Yeah.
But you know, it doesn't need to be the same motif. They don't. I mean, they don't need to be enriched. But what you can ask. I mean, we now have such a rich dictionary of regulatory motifs. You can ask, hey, are they, you know, to the right of the curve? What about the bimodality? Have you tried any sequence analysis there to basically see what distinguishes the randoms in one mode versus the randoms in the other mode?
Yeah, that's also ongoing. Right now we have, I'm trying to use, for example, the string kernel SVMs to find k-mers that might distinguish between these two. I've thrown these sequences through the meme suite of tools and things like that. So far, nothing has come out
Yeah, but you
yet.
don't need to rediscover them. You can just go take the existing ones and check.
And scan those.
And the last question is, actually, why don't you go ahead?
Okay.
I mean, so one problem I have with this assay is the fact when you put in a binding site that was a binding site in the other part of the genome, a negative result tells you nothing, right? So how do you deal with that? You see a modest enrichment, so you're taking it completely out of its chromatin context from somewhere else in the genome, putting it into this reporter assay, and a negative control tells you nothing about it. So how do you deal with that?
Yeah, I guess I'm not sure I understood. The test is that we have these CRX motifs that are throughout all the genome, and we're testing them in a cell type where CRX drives much of the major transcription. So we put it in a reporter assay.
Yeah, it could be, like I'm saying, it could be functional at parts of the genome. It's just that a negative control in your plasmid assay means nothing then, right?
Yeah, but so I'm taking non-functional sequences from the genome that were not bound
But a substantial part of your distribution overlaps the random control, right?
That is true, yeah.
Somewhat naive question. Are there any chromatin features that are happening on your reporters, like for instance DNA methylation, that could affect transcription factor binding? Or
That we would love to do. So we are making some plans, they're still early, to test some of these chromatin marks and other things on the plasmids. We don't know how they're chromatinized. And so I'd be very excited to see that result. I'm not opposed. I think my message is locally encoded sequence, but it could work through chromatin at that level.
Terrific. Well, please join me in thanking Michael and all the rest of the speakers from this session.